to me that I should do a little intro here as we've got more and more people coming in and joining us. This is so exciting. For those of you who don't know, I'm Dr. Nicole Beurkens. I am a pediatric clinical psychologist and nutrition specialist doing these clinical grand rounds with Microbiome Labs. And we have Kiran Krishnan, co-founder of Microbiome Labs and microbiologist with us. Kiran did the talk for clinical grand rounds earlier this month on this topic of ACE2 receptors and underlying inflammation and COVID-19. So this is an opportunity for all of you to hear more about that by getting specific questions answered on that topic, or also other topics. We have lots of a wide variety of things coming in. So really excited that you all are with us. Kiran and I are both super excited about doing these clinical grand rounds, and it's great that there's been such a wonderful response to them. Okay, so Kiran, let's go to, Kim asked if you could just give a brief review of what you talked about earlier in the month on the actual session that we did of the ACE2 connection with COVID-19. She said, I want to understand why that can make our bodies seem to be more susceptible to COVID and just to have a better understanding of that. So maybe just a brief review.
Yeah, and it's purely because like any other virus, the virus requires a target. It requires a receptor on our cells in order to bind and then activate its ability to transfer its genetic material into the cell. This particular virus, like many other viruses, especially in the corona family, use that ACE2 receptor that is pretty ubiquitous in our system. The virus has evolved to find and utilize ubiquitous receptors so that it has many chances of infecting the body, because depending on where it enters in the body, if it enters into a tissue that doesn't express its target receptor, then it has no chance of multiplying and actually hijacking the host cellular machinery like it wants to do. So the ACE2 receptor is a very ubiquitous receptor that's expressed in lots of different tissues. So no matter where it enters in, whether it's through the mouth and going into the digestive tract, in the upper respiratory tract, getting into the lower respiratory tract, it doesn't seem to matter, because then it has its target available to it. So the spike protein on the virus basically binds to the ACE2 receptor. There is an enzymatic proteolytic activation that occurs because of that, and then that allows the virus then to penetrate through and or inject through its RNA that then starts taking over the cellular machinery.
Awesome, great. Annette is wondering about specific applications of megaspore biotic and COVID-19, both in being proactive. Is there a role for that in being proactive if somebody hasn't had the diagnosis of that, and also if somebody has been diagnosed, if someone is in the process of having an active illness or recovering from that?
Yeah. So the biggest impact of that megaspore is going to have as it relates to COVID-19 is the likely reduction in the expression of ACE2 throughout the body. And the reason I say that, and mind you, we have not done a specific trial showing taking megaspore reduces ACE2 expression, where it's using the transitive property of the impact of LPS, because one of the big drivers of ACE2 expression in many tissues in the body is the presence of lipopolysaccharide in circulation. And in fact, the way your body combats the damage that LPS does is by increasing the expression of ACE2 receptors in that tissue, right? So it's not a big scientific leap or a big guess to say that if you reduce the amount of LPS and all the sequential cytokines that go along with that, from circulating in your body, you're going to reduce the number of ACE2 expressing cells in your system. And that's throughout the body, because there's evidence that shows that LPS from the gut can exacerbate inflammation and ACE2 expression in the lungs as well. So it impacts all parts of the body. So our position is we've got to bring down the inflammatory status of the body. We've got to bring down the reasons for expressing ACE2, which then provides a target to the virus. And we got to bring down the inflammatory impact on the immune system, because inevitably, once the immune system detects the presence of the virus, it's going to go ramping up its inflammatory response. So if you imagine your basal inflammatory response is here, your gut is leaky and you've got kind of this elevated basal inflammatory response, so you're now here. And then the virus comes in and you're going from here then to up here, which becomes dangerous, rather than from here to here. So our baseline is what we're saying is a big benefit and something to keep in mind as well. Now, the other aspect of it that's really important to us is, increasing diversity in the microbiome certainly helps the immune system function. There's evidence that shows that the increase in diversity in the microbiome increases the function of T helper cells, T regulatory cells. These are all really important cells in the immune response towards the virus in order to negate the whole cytokine storm type of response. So, those connections are pretty clear, that diversity in the microbiome, reducing the permeability, all of that gives the immune system a better way of functioning and a better chance then of having, hopefully a favorable response to encountering this virus. And, we're not, you guys are all practitioners and doctors and so on. We're not saying that we have any evidence that it treats the condition. That's not the premise we have. What we're looking at is how do you put the immune system in the body in a better space, in a better condition to deal with it should you encounter it.
Right, absolutely. So following up on that, I'm going to summarize a bunch of questions that have come in around specifically using the products, Megaspore Biotic in particular, and dosing as it pertains to somebody who has an active illness, somebody who's been diagnosed or it's assumed that they have COVID-19. Lots of questions coming in of, do we increase dose? Do we leave it the same? Should we start somebody on, just the regular dose if they haven't been on it before? Obviously, as you're saying, we don't have any, clinical studies on this. This is all just really about supporting healthy immune function, but I'm just going to let you comment on that because that will address a number of things that have been coming through.
Yeah, so if it's a new patient right now that has never taken Megaspore, let's assume they don't have an active infection, because if they have an active infection, there's lots of complications and they're probably either in a hospital, or if you are their primary care doctor and you've made the assessment that they can recover from this at home, you're working with them on home-based recoveries. But if they're a new patient, we would, no matter what, still taper the dose up for them. But the taper in most cases can be pretty short. We're talking about a taper to the full dose of 2 caps within the first week or two. You don't have to go really, really slow. You can start everybody with 1 capsule a day, and then after 2 or 3 days, go to the 2 capsules a day. If it for some reason becomes too much for them, you can go back to the 1 capsule a day for another few days, and then try again at the 2 capsule a day dose. So you could try to get that tapering into the normal 2 cap a day dose within the first 7 to 10 days. But we would still recommend, if they are a new patient who's never taken it, do it at the tapering level. If they're an existing patient who's always been taking it, you could totally keep them at the same level. You can go up higher if you want. We always encourage people, if they're taking 2 caps a day and that's their normal amount and that's what really helps them and keeps them in control and check, should they come across any sort of infection, and it doesn't have to do with COVID-19, if they get a cold or a flu, normal cold or flu, we tell them you can bump it up to 3 or 4 caps a day as well. That's what I do personally as well. I do that for my kids as well. They typically take 1 cap a day. They're 6 and 9 years old. Should they get the sniffles or rotavirus or something, we go up to 2 caps a day. So we essentially double their dose for a short period of time. So new patients, no matter what, taper them up still. Existing patients, if they're at 2 cap a day and you want to bump them up because they're starting to feel not well, you could totally do that without a taper.
Okay, excellent. Would the same apply then? Some people are asking about Mega IgG. Same thing. If they're new to it, start them with the regular dose. And how would you encourage people to think about that?
Yeah, the Mega IgG is a 4 cap dose. You can take upwards of 8 capsules a day. I take 4 at my first meal and 4 at night before bed is what my regular dose is. The mega IgG typically doesn't have any sort of die-off or Herxheimer reaction. It's really rare that we ever hear anything about that. So there's no issue starting them with the 4 cap a day dose. That's going to be extremely supportive of their immune system. There's studies with this particular IgG in HIV enteropathy and seeing the really favorable immunomodulation even under those conditions. So this is going to be a really important product for anyone at this time, especially. And you don't have to go lower than the 4 cap a day dose. You're always welcome after they've done the 4 cap a day dose for a couple of days to have them try the higher dose if you think they need it, but you don't have to step up to it.
Okay. Awesome. That, I think, covers the majority of the questions that came through really about the COVID-19 situation. Some more may come in, so we can jump back to that. But let's get to some other great questions coming in here about certain conditions or the products in general. So Kiran, someone is asking how to decide whether to use the Megaspore or the HU58 or both. Like, what are the different things that a practitioner should be considering there?
Yeah, so the Megaspore is always the starting point for people, right? No matter what you're seeing, the Megaspore is the baseline. That's what's giving you all of these kind of baseline microbiome improvement factors. The time when you think about adding in the HU58 would be if you suspect some specific infection in the patient's gut. They've had a history of C. diff, for example. They don't have a current active C. diff infection, but they've had history of it. Or you suspect from previous tests that they may have an opportunistic or other type of pathogen in the gut that's causing some of their symptomology. So if you feel strongly about wanting to bring control to potential pathogens in the gut, that's when you would add in the HU58. The other time you would do it is if they are coming to you and they're starting on an antibiotic or have recently been on an antibiotic. Because remember, antibiotics, if you take them, even a 7-day dose, the effects of the dysbiotic effects of that antibiotic can be present up to 2 years after the course of the antibiotic. So even they might have come to you and said, oh yeah, I had a number of antibiotic courses 4 months ago. Even though it was a while ago, you could still treat them as if they are currently on the antibiotic because those dysfunctional effects last for so long, and you do need to go in there and fix it. Our antibiotic protocol includes Megaspore and HU58, and in the case of an active use of antibiotics, we add in the Restore Flora as well, which has the Saccharomyces boulardii in it. Although if the antibiotic use is in the recent past, then the Megaspore and the HU58 together are fine. And again, if you suspect something is causing infectious-like response in them, then you could throw in the HU58 to start, and as a way of kind of going after it. And the reason is the HU58 is the subtilis that's in Megaspore, but it's at a higher dose. And the subtilis of all the strains we work with is a very powerful competitive excluding bacteria, meaning it's really good at finding problematic bacteria and bringing down their levels. So really what you're doing is you're just giving them extra amount of the subtilis. And if you're tapering them on the Megaspore, you could start with 1 capsule of the Megaspore and still do 1 capsule of the HU58 from the start, and then continue the taper of the Megaspore, but keep them on the 1 cap of HU58 a day. Ideally, what you want them to be on is 2 caps of Megaspore and 2 caps of HU58, BID essentially.
Mm-hmm.
So that would be kind of the goal and how you think about it. So think about the HU58 as kind of like a intelligent antimicrobial. If you feel an antimicrobial is needed for that patient's condition, then try the HU58 first with the Megaspore.
Awesome. Fantastic. Okay. And you answered a bunch of the follow-up questions about that too, so that's great. Michelle is asking, are there any clinical studies going on right now, or are you thinking about any in the future regarding the Microbiome Labs products and biofilms?
Yeah. So we are. That's one of our engagements with Sacramento State. One of the things that really interested us in Dr. Rob Crawford that runs the Microbiome Lab there is his postdoctoral work was all in biofilms. And here's the thing, biofilms tend to get a bad name, but they're not. Biofilms are extremely important because the vast majority of good bacteria in the gut also live within biofilms, right? So biofilms are, just think of them as kind of the lattice or the ecosystem in which bacteria live. They also use them for communication, and there's lots of function to biofilms. So the idea that biofilms are bad, they're associated with pathogens, is not right. Most of the good bacteria in your gut also live in biofilms. So just the target of just trying to destroy biofilms can cause more adverse effects than you really want to, because it's also going to harm the complex in which your good bacteria live, and protect one another, right? So that's the important message here. What we are trying to study with Sacramento State is, what is that communication and relationship between the various types of bacteria within a biofilm itself? We know that Bacillus has the capability of breaking down other bacteria's biofilms, and in part that may be how they go after certain pathogens, because Bacillus has the ability of producing an alpha amylase enzyme that just breaks down biofilm. So if they detect there's a pathogenic bacteria in a biofilm, they know how to go after those bacteria, and there's studies to show they are effective at competitively excluding bacteria that are known to be biofilm producers. So that's the extent of what we're doing in the biofilm world. We really want to just kind of understand the bacterial ecology, the communication, and how all of that is structured within a biofilm. We're not looking at biofilm disruptors and so on, because we think we don't know enough about biofilms to go in and start busting them all up non-specifically, right? So that's the bit on biofilms.
All right, fantastic. Okay, and while we're talking about potential research or projects that you're thinking about, Laura Lee has a question about whether the company has any studies looking at sporebiotics' ability to reduce things like P. gingivitis, T. denticola, another one that I don't even know how to say, sorry, Laura Lee, or the red complex bacteria in the oral cavity. Any studies done to date, or any plans to look at studies on those kinds of things?
Yeah, so we actually have completed, I think, 21 patients or so on a oral microbiome study right now. We were looking at a combination of the changes in the mitochondria in the gingival tissue, where we're looking at, so which means we're looking at kind of the health of the gingival tissue. We're looking at bleeding on probing, which is a way of measuring the inflammation and the gingivitis component of it. And then we also did oral swabs to look at changes to the oral microbiome. We don't have the oral microbiome data yet on this first small subset, and this is, we're aiming for this to be kind of a 50-60 patient study, but we had never done one like this, and there isn't enough data to really understand how we need to power the study. So we've taken this initial cohort and we're trying to analyze the data. So that actually has been done, and we're waiting on the analysis from the microbiome side. We're hoping to see that there is a measurable change, because there are studies that correlate dysfunction in the gut to dysfunction then in the gingival tissue in the mouth. And so our hypothesis is that if we can then improve the gut microbiome, which we know we are doing with the spores already in other studies, then will that translate to improvement in the mouth as well. So that's where we are with it. I don't have the data yet, but we have done that study. I'm hoping we'll have some sort of analysis on this initial part of the data, in the next month or so. The problem is with 20 patients, so that might be too small to see a measurable difference. And that part we're not sure either. If you're not familiar with clinical trials, when you look at different endpoints, especially if you're looking at your primary endpoint or a secondary endpoint, you have to power the study for that endpoint, which means there's a statistical analysis based on previous studies of you run through the stats and you go, okay, to find significant difference, if there's going to be a significant difference in this particular endpoint, you need to power your study with 80 patients. That's that kind of power analysis we didn't have at our disposal, because this has never really been done with probiotics. So we're just kind of looking at the first 20 patients, and we may not see any measurable difference in that, and that might give us the impetus to say, maybe we're not changing anything, or it might tell us, we are, depending on how the patient, other factors in the mouth, we might decide that if we did 20 more or 40 more, we might see more measurable changes. So it's ongoing. It's a good question. And that's a really interesting area for us as well. And here's the other reason why this is an interesting area for us, because not only is that obviously good for oral health, but oral dysbiosis to me is a big driver of gut dysbiosis, and then also inflammation in the body, because a lot of those bacteria, especially ones that are LPS producers, can directly dump into the blood through the oral cavity. So it's an important area to focus on.
Fantastic. And so many great studies that, you guys have in the works and are planning. So great that people are asking about that. Let's go to a couple questions specific to the total gut restoration system. Mm-hmm. So I think we can combine a couple here together. Kelly is asking, in regards to that total gut restoration system, is it always necessary to wait to take the prebiotic and the mega mucosa, or is it okay for patients to begin taking the probiotic and others at the same time? And then James has a related question about using the total gut restoration program with women who are breastfeeding.
Yeah, it is fine to start at the same time. We do prefer that they get on the megaspore first and get through that tapering period, right? So if they are one of those patients that you can kind of taper pretty fast, you can get them to the full 2 cap a day dose, let's say within a week, then at that point, you can totally start them on the prebiotic and the mega mucosa. Most of us that work at Microbiome Labs, that's kind of how we all did it. But we were all also already on the spores to begin with. So you don't necessarily have to go through that cycle. That cycle is really for like a real stepwise rebuilding of the gut. It doesn't mean that you can't do it all kind of right away at once, which would be kind of the month 3 scenario right off the bat. But we do ask that you first taper them up on the megaspore so they get their system used to the megaspore itself, and used to the changes that are going to inevitably be happening in the gut because of megaspore. And once they're kind of used and comfortable with that, then you could definitely throw in the prebiotic and mega mucosa. With regards to using it with pregnant women, absolutely. There's or nursing mothers, I think was a question.
Yeah. Yes.
But let's address both. Pregnant women, absolutely, because of course you want mom's microbiome as healthy as possible, because she's going to be passing that on to the kids. So anything that's going to help the diversity, the presence of keystone strains, the reduction in endotoxemia is going to be important for mom, and then thus by the transitive property to kids, to the child during birth and then right shortly after birth. With nursing moms as well, there is no reason that you wouldn't be able to do it. There's no specific contraindication. The mom's mammary glands get loaded with her gut bacteria, and that's part of how she transfers bacteria to the child through milk. So the presumption is the healthier her gut is, the lower her inflammation is, the better off her composition is in the breast milk, and thus the child actually gets more benefit. So there's no reason to think that it's not safe for women who are nursing. Now, mind you, on every product, supplement product essentially put out, you have to put a nursing pregnant warning on there. That's just standard, right? Whether there's risk or no risk, that's just standard what you do from a regulatory standpoint.
Mm-hmm. Great. A question also related to this, which I think you touched on, but just so that we're clear with people about it. Lindsay's asking, can you talk about how and when you've got the spore probiotic and then you're thinking about adding or moving forward to the prebiotic powder and then to the Restore Flora, how you think about adding those in?
So as part of the TGR, you mean?
Yes. Yeah.
Yeah. So the easiest way to do it is obviously you take the megaspore as a capsule. So you're taking both capsules together with any meal of the day. The way I do it when I'm using both, and I don't always use both, I'll go through periods because I've been using them for some time now. I do the mega mucosa basically every day. That to me really helps my system, and we kind of see that across the board because mega mucosa is actually the second best-selling product in our total portfolio next to Megaspore. So that seems to be across the board. People are finding that megamucosa is like a really important and effective maintenance for them for various conditions and reasons. We've also heard lots and lots of testimonials how they saw, a real measurable improvement in their condition with just the Megaspore, and then when they brought in the megamucosa, it just really was the tipping point for them to get better. So you could do that every day. There's no issue with that at all. If you were for the first time going through the cycle, what you would do is you would start with about half a scoop of the mega prebiotic and mix it into a large bottle. Like this is my normal shaker bottle. We do also have Microbiome Labs shaker bottles, which are in the dishwasher right now, but with about this much water. I would say this is maybe like 16, 18 ounces. I do about half a scoop, and then I mix it up, sometime in the afternoon, right around my first meal, and then I'm sipping this throughout the day. We recommend with the prebiotic, it's good to sip it throughout the day rather than taking it all at once, just mixing in a glass of water and dousing it. There is some data that with certain types of carbohydrates and oligosaccharides, there's a benefit to incremental dosing of it throughout the day. You get better utilization of it. So that's how we recommend it. If you're taking both at the same time, the megamucosa and megapre, then in that first mix of the day, you add the full dose of megamucosa, about half a scoop of megapre, and then you sip that throughout the day. In the evening period, you can add the other half of the megapre scoop and sip that the rest of the evening as well. So then you're getting good hydration, you're drinking liquids, and you're getting it in throughout the day to help you out. When I do both, I actually just go full on 1 scoop of megapre, 1 scoop of mega mucosa in my first bottle, mix it up. I'll sip it when the liquid in the bottle gets down to about here, little above half. I just refill it with more water and shake it up and keep drinking it throughout the day. So this 1 bottle, I just kind of keep refilling it and drinking it throughout the day. So then the taste gets more and more diluted as you go through the day, but I'm getting lots of hydration. I'm getting lots of the active ingredient in.
Fantastic. Yeah, and I think it's helpful just to know how different people use the products and actually incorporate them into the day, because obviously it's one thing to recommend to clients and patients what to do, and but it's another thing to help them think about how to manage taking all that stuff. So I think that's super helpful.
Yeah.
Susan Wester has a question. I'm not sure if this is one that you have an answer to, but I'll ask it anyway. Going back to some of the information that we're talking about with COVID, she says, I read that approximately 30% of people who had COVID-19 will develop the antibodies, and the remainder of the 70% simply had innate immune response, which did not produce any antibodies. Is this possible? Is this true? Is it just too soon to know any of this?
Yeah, it's a little too soon because we're still calling into question the accuracy of the antibody tests, right? But let's take just a hypothetical situation, for example, that is true. What would that mean? It would mean that they had IgA antibodies, which are ones that are produced that are not as specific as IgM and IgG. That they had IgA antibodies that actually happen to fit components of the virus and took care of a lot of the viral load. Or actually I would say they also amplified their IgM response and did not get adequate elevation of the T helper cells that go from an IgM to an IgG response, right? So maybe the infection was sequestered between IgA and IgM. IgM is more specific than IgA, and it's a very powerful neutralizing antibody. It's a large antibody that basically neutralizes the virus. Now, the thing with IgM is it doesn't last very long. So the patient could have, gone through COVID and between IgA and IgM, dealt with the infection and never got the stimulus or the impetus to develop the IgG B cells against the virus, and maybe in part because it was managed quickly enough where you didn't have that proliferation of the antibody producing cells for IgG antibodies. And then if you try to detect antibodies in that patient that are specific to COVID, let's say 2 months, 2 weeks, 3 weeks later, you may not find any because the IgA antibodies are not specific to COVID. They're going to be non-specific ones that can happen to bind to different things. And the more COVID-specific IgM antibodies would typically be gone at that point. IgM, in many people doesn't last much more than a couple of weeks. So that may be a scenario in which you could have that. But I would say that our antibody data right now is really tenuous because we still don't know how accurate most of those antibody tests are. And we just don't have like the robustness of the data to kind of hang our hats on it.
Yeah. Okay, great. So let's go to, again, trying to group questions together here. Several questions from clinicians about how to manage it if clients or patients are experiencing significant constipation when they start on either the Megaspore biotic or someone even mentioned the Megamucosa, basically any of the products. If you have somebody who's experiencing significant constipation, what would be the best course of action there?
Yeah. So we've seen it managed and resolved in two ways. One is they actually kind of punch through it, where you actually increase the dose, and that's what is needed to get them over the hump, and then they start having good and normal bowel movements. In other cases, you actually have to taper back the dose quite a bit and go longer at a lower dose to manage that constipation. So it kind of depends on how your patient is responding to it and how comfortable they are with testing a higher dose. So for example, like people I personally know that start taking it, we do see about 10% of people responding this way, where they do get an increase in constipation. If it was somebody I knew, it's just a friend or family member, they started taking it and they call me and they go, hey, I'm at 2 caps and I feel like I'm constipated and my bowels slowed down. It's been a couple of days, or I'm at 1 cap. I would say, you know what, just punch up to 2 caps and see what happens. Oftentimes, going up to the 2 cap level would be what is needed to kind of break through that barrier. If they're willing to do that, that's how I'd advise them to do it. Tom, for example, will more than likely be err on the cautious side, and he'll say, you know what, just back down then. If 1 capsule is too much, just back down to half a capsule and do that for another few days, and then see if it changes the bowel movement. In most cases, we see this kind of constipation not last more than 4 or 5 days. So it really kind of depends on the psyche of your patient, how willing they are to try to punch through it. And if they really want more comfort, a gentler approach, then you would just taper them back down to half the dose where they felt the constipation, and just let that go for a little longer till the bowel movements start changing and improving.
Okay, great. Question that came through, I'm trying to find it here on the chat box, but I think I remember what it was. If somebody's working with a baby who is not being breastfed, who's being formula fed, they're asking about the appropriateness of opening the Megaspore biotic capsules into formula for babies or for toddlers.
Yeah, there shouldn't be any issue of opening it up and adding it to the formula right before you're going to feed them. I wouldn't add it in and then leave it in the formula for a period of time, because the bacteria could start fermenting the formula, although that risk is dramatically reduced if you put the formula in the fridge at refrigeration temperatures. But still, in that case, I would say no. If you're going to add it into the formula, you could do it. You should do it right before the baby's going to feed. Now, most babies who are not yet eating solid food, the way we recommend them dose is just you open up the capsule, you put about half or a quarter of the powder on any in a plate or something, and you just take a pinch with your finger and put it in their mouth. That's the best and easiest way right before feeding. So then it's in their mouth. A lot of babies actually kind of like that interesting texture in the mouth, and then you feed them formula, and it kind of washes it all down into their gut. So that is the kind of the preferred way of doing it for babies, rather than needing to mix it into formula. They just need a pinch in that beginning period, in the first 6 months or so. After about 6 months, you can give them much more uniform half or 1 capsule a day, but up to that point, a pinch will go a long way.
Okay, excellent. Okay, Katie is asking about Mega Myco and wondering about if there's the potential for it over time to have a negative effect on the microbiome. She says she's heard that you can become allergic to propolis if you stay on it for too long. How many consecutive bottles of this product is it safe to be on? Is that something I need to be concerned about?
Yeah, so development of allergy to anything, whether it's propolis or any other compound, it's not necessarily a factor of a duration of exposure to it, right? It really implies that there is a tendency for development of sensitivity within that person's immune system or microbiome and so on. So there is no tenant in immunology that X amount of duration of exposure to an antigen causes sensitivity. It's really the exposure to the antigen and then the dysfunction of the immune response towards that antigen that causes the sensitivity. We've not seen any studies that show that there's a particular risk of developing sensitivity to propolis. So if that has happened, if you heard about a case from another practitioner or so on, that's likely because of whatever else was going on in that patient's system. It's not the propolis itself. Now, I would also say that if you're dealing with candida, or other fungal issues, which is the purpose of that product, you should hopefully be able to get that condition under control in a 4 to 6 week period. If you're using kind of a full dose of the product, if you're at the 5 to 10 capsule a day, or in some cases even upwards of 15. If the patient has had a hard time scaling up, we've had people that do really well and feel really good until they get to like 8, and then they can't go above 8 in a day. Then it becomes a little too much. So then they have to stay at that 7 level for a longer period of time. We've not seen any issue with people having to stay on it for longer periods of time to bring the issue under control, but it's also not something that they should be having to take indefinitely, right? There should be a time period on it, because if you're not bringing the condition under control within a certain amount of time, then there are probably other things that need to be thought through. Most often with people with fungal issues, if they're starting the Mega Microbalance for the fungal issue, they're also hopefully using the Megaspore and maybe even the HU58, depending on the complexity of the issue. They should be able to bring down and control that fungal issue within the first 6 to 8 weeks. And then after that, they shouldn't need the Mega Microbalance again.
Fantastic. That answered a couple of other questions that had come in too. So great
I'm
job.
knocking them off 2 or 3 at a time. Look at that.
I know, I love it. Grouping a couple others together that have come in related to PPI use. So proton pump inhibitors, maybe for patients who have had, chronic reflux issues, GERD, those kinds of things. So just to summarize the questions around that, in using the total gut restoration program with that, is there a certain protocol to be thinking about there, and particularly in regards to thinking about how to wean off of the PPI in using some of these products?
Yeah, the most success we've heard clinically on weaning off of the PPIs comes with the use of MegaGuard. So MegaGuard is the artichoke in there is indicated in Europe as a treatment for reflux, and so it can be quite effective in doing that. And so, if you're trying to work with your patient to wean off the PPIs, you'll have to make sure that they're also on the MegaGuard product. We've had conditions where they can just swap it 1-to-1 in one day. They go, "Oh, I'm starting MegaGuard, I'm stopping the PPIs," and they feel fine. So you'll have to play around with that to see. You probably start them at this, they're on the PPI, and then you start them with the MegaGuard, and then you slowly kind of taper down the dose of the PPI while keeping the MegaGuard, and then seeing where they are, what threshold they might have. Where they at this particular dose, then all of a sudden their symptoms kind of start to come back, and so you want to go a little bit above that and keep them both together at the same time and keep testing the reduction of the PPI. Now, with regards to TGR, you wouldn't change anything about the TGR. The TGR is going to do what it has to do in fixing the leakiness in the gut and the diversity issues and the dysbiosis and so on. none of that has to be impacted by the presence of PPIs. We do have, reports that just by fixing the gut using the megaspore and so on, that people have been able to reduce their PPIs. But I would say to really be effective about that, the MegaGuard is going to be important to add in there.
Okay, fantastic. Very, very helpful. Wanted to address again a couple things that came in, but just to summarize the issue of why is it that some companies recommend that you only use spore-based products for a limited amount of time versus what you recommend?
Yeah, my best response to that is because they really don't understand spore-based probiotics, right? You'll have to remember that we were really the pioneers in this, bringing this out into the marketplace. So most of the companies and people are quite a bit behind on it. They don't really understand it, and also it's a marketing position, right? For the most part, loads of our practitioners have found that because they're using the spore-based probiotics, they really don't need anything else. Between the spore-based probiotics and diet changes and other lifestyle modifications that they would get their patients to make, you don't need a lot of these kind of multi-strain probiotics anymore, these what I call kitchen sink probiotics. So some of those are seeing reductions in use and volume, and and then they're trying to figure out a competitive position, and so then their position is, wait a minute, okay, spore-based probiotics might be effective for a period of time, but you shouldn't use them for a long period of time. Why? I've never seen any reason for that. I mean, if you look at nature, which is where we typically get these bacteria, these bacteria are ubiquitous in the natural environment. They are commensal bacteria in the gut, which has been verified by a couple of published studies as far back as 2009. And in nature, we would be getting them every single day. We'd be eating them with our food. We'd be picking them up from the environment. And they are transient in that they go in, they spend a bit of time fixing up the gut, and then a good portion of them leaves, and they kind of maintain this 10 to the 6 CFU level in the gut, in various portions of the gut. So there is no reason that we can think that's reasonable why you would hold a position that you shouldn't use them for long term. I've been using Megaspore now for 7 years, and my daily dose is somewhere around 4 to 6. Because I travel, or used to travel a lot, and I put my body through a lot of stress, and I find that the higher dose works better for me. But we've had tens of thousands of patients since day one who've been taking it. And again, it matches what happens in nature. To me, that's a very important part. Where in nature do you get 100 billion CFUs of lactobacillus and bifidobacteria every day? You don't, right? Those are the commensal bacteria that we typically get one shot from mom, and some from dad as well during the birthing period, and breastfeeding. But outside of that, it's not normal to have that level of exposure to those types of strains. But it is perfectly normal and reasonable to get constant exposure to spore-based bacteria that are using the environment as a vector to get in.
Great. Just want to let everybody know we're going to be able to get to a couple more here, and then we're going to need to wrap up for today. But those of you who have had questions in the chat box that hadn't been answered, this is great. This is helping us to know what kinds of questions and topics you all are interested in. I'm taking notes, learning and development team's taking notes on all this. We'll get some speakers in. This is really good for us to be able to address your most pressing needs as we move forward with these clinical grand rounds. So even though we won't have time to get to very many more questions, go ahead and continue to put them in, because we will save that information.
Yeah, and maybe one of the things we can do, Nicole, is we can extract those questions and answer them by typing them, and then sending out the Q&A typed out to the group.
Yep, right? we can. There's a lot of questions, Kiran. A lot of questions. We've got somebody who started a screen sharing here. Let's see how we can get off of that. All right. Hey, Linda, you somehow clicked on screen sharing, and I need to figure out how to stop that here. Kiran, question about while I'm figuring that out, question about dogs. So for those of our people in the chat room who are working with animals, they're wondering about using the products with animals. Specifically, there was a question, there we go, a question about the Megamyco with dogs. And some of the other products to use, and the HU58 to use with dogs or pets in particular. Any thoughts on that?
Yeah, I mean, so we've had people using Megaspore HU58 with dogs and pets for a long time now, but we've also now gone ahead and developed an actual dog and pet probiotic called Phytospore. And we did and conducted the first of its kind clinical study showing that pets also get leaky gut, or in this case, dogs get leaky gut, and the leaky gut is consequential to them because it is a big source of chronic low-grade inflammation. And then we also created a formula using a couple of the spores and Pediococcus, and we showed in a second study that we were able to reduce that leaky gut and all the inflammatory markers as a result in a significant way. And both of those studies have been submitted for publication. Hopefully we'll publish sometime in the next few months. But that formula is Bacillus subtilis, the HU58. It is Bacillus ligniformis and Pediococcus acidolactici. All three are known commensal organisms in dogs and in cats as well. And highly effective. They're some of those strains like ligniformis and subtilis are used in animals across the board in feed, certain types of feed scenarios. So yeah, you don't have to guess anymore. You can use Phytospore, which is the pet-based probiotic. You can use it on dogs and cats, and it will stop their leaky gut.
Awesome. Okay, I think we'll get to just one more here. Let's do, I had just seen one. Okay, why don't we end with a COVID related question too, since those are the times we're in. We started there, why don't we wrap up there today? And again, thank you everybody for all of your questions here. We're gonna end with this one from Kelly. She's wondering if you can talk a bit about Th1 versus Th2 immunity, how they might play into COVID-19, and how or if spore-based probiotics might help with this, particularly in autoimmune patients. Any thoughts on that?
Yeah, so and when it comes to autoimmune, then you also have to consider Th17. That's the real culprit when it comes to autoimmune, right? So let's address autoimmune first. Autoimmune tends to have a overexpression of Th17 in particular, driven by interleukin 17 to begin with, but then you see an upregulation of the T helper cells that are categorized as Th17 that then facilitate the formation of these B cells that can make self antibodies. So when it comes to autoimmune disease, the key is to bring down the Th17 expression. And one of the ways that Th17 is brought down is by the upregulation of IL-10 and the upregulation of Treg, the FOXP3 positive Treg. What we do see, and we've seen this in one of the studies we've published, and we're working on it right now in other mechanistic studies, is we do see an upregulation of IL-10 when you use the spores, which is balancing out the Th17 upregulation in autoimmune. There is also evidence already that Bacillus endospores upregulate Treg expression too. So it's the regulatory part of the immune system that needs to get amplified in order to come and dampen that Th17 autoimmune type response. Now with Th1 and Th2, both are really important. Early on when I started working in this functional medicine space with functional medicine practitioners, we would always get kind of this one is bad, one is good mindset, but both are equally important in the body, and the key is being able to shuttle between the two depending on what the immune system needs to take care of. You of course need to move towards an adaptive immune response, so you do need that Th shift going on between 1 and 2. The question is, how do you facilitate that shift and that modulation? That's all done through T helper cells. So T helper cells are upregulated when you have probiotics, or when you have an improvement in your microbiome, like an increase in diversity in the microbiome, and the spores have also been able to show to help modulate that balance between Th1 and Th2. So it's not one or the other. Both have to be present. Both have to be functioning. It's, do we have a way of modulating and balancing between the two? And in general, the spores seem to upregulate cytokines and the T helper cells that are required to modulate between the two. So hopefully that help. But with the autoimmune side of things, it really is focused on bringing down the Th17 by upregulating the Treg system, right? So with COVID, your body's going to flip between the two depending on the kinetics of your immune response to it. Like we said, some of this early information is that some people don't end up getting an IgG response. So maybe they don't have a strong Th2 response, or maybe they don't have a strong Th1 response, but they're getting more of a Th2 response against COVID. We still don't know enough about it. But what we do know is lymphocyte proliferation becomes important. What we do know is reducing inflammatory response becomes really important, because we want to do whatever we can to negate that cytokine storm. We also do know that if we can bring down the inflammatory load in the immune system, that should help with the way your body's dealing with it. So that's the most we can say, because we really don't know a lot about the kinetics of how the immune system responds to COVID in the first place.
Awesome. Great information. Kiran, we thank you so much for taking the time to be with us and to do this today. Thank you to the hundreds of you that joined us for this. Really excited that you're excited about being a part of these. We've captured all the questions. That'll help us as we move forward with providing the best speakers and information to you. Be on the lookout. The first Friday in April, we will have our next Clinical Grand Round session available to be emailed to all of you. And then the final Friday of next month, we will, May, yeah, that's right, next month is May, we will have another live Q&A with the speaker from May, and be able to answer lots of your general questions as well. So thank you so much for being here, and we will see you next time. Take care. Bye-bye, everybody.